as I said, today's outstanding speaker is Professor Roberto Verzicco. I would say a few words about his career. He got his PhD from Sapienza University, and then he moved to Polytechnic University of Bari. Then he moved back to Rome to Tor Vergata University. He also has appointments with the University of Twente and with the GSSI, as you have seen. Roberto, I should say, is one of the leading figures in fluid dynamics in Italy, for sure, and worldwide, I would say. He has given a lot of important contributions to the numerical simulations of turbulent flows and complex flows. He has developed a special interest for biological flows, for instance, the stuff that is going to show you today, which is really impressive, I would say. Among other distinctions, I would say that Roberto is a fellow of the American Physical Society, of the EUROMECH, and he was the recipient of the High-Fidelity Award from the American Physical Society. Among other things, he's also associate editor of the Journal of Fluid Mechanics. He has published 2 papers in the annual review of fluid mechanics. An incredible track record in terms of publications. I'm sure I'm forgetting something, but I think in the interest of saving time, I'll leave the floor to Roberto. And today is going to talk about HPC model of the human heart, which is, I think, a topic that we are all interested in. Roberto, floor is yours.
Thank you. Welcome. Thank you for being here. So in the next 1 hour and a half, more or less, I will talk about the fluid mechanics of the heart and the tissue mechanics and the electrophysiology and some of the other problems we are trying to solve. So since you know that any scientist is obliged to be fair and to say the truth, I should already start with something. So I was not the original invited for the lecture. The original one was Detlef Lose, but he had some family duties. And then today you have the second choice, but you will have anyway Detlef Lose on Tuesday, is it? Yeah, so this is just to unveil everything. The second point is that I'm the speaker, so you see me as the visible element of the group, but many of the things I'll be talking about have been done by Francesco Viola, who is sitting there, and certainly without him, what I'll be showing would not have been possible. So this problem is a huge undertaking, this project. It is something we have been working at least over the last 10 years. And so over the years, we have collected many many collaborators. And here I'm showing you a list, hopefully. Okay, here it is. A list of collaborators, and PhD students that have been working. So just to get you oriented, those in black and white are GSSI students or former students. All the others are people who we have collected over the years. Let me see if I can move this part. Okay. And this one? No. Okay, that is okay. So there are some colleagues, some former students, and of course here I'm not mentioning the countless master students, bachelor students who have been making troubles and bringing enthusiasm in the lab and always keeping up with the problem. Good. Now, the field has a huge literature. If you try to collect even the most basic papers for a single of the topics I'll be talking about, you would take, say, a month to collect everything. So if you want to have a shortcut, you can refer to this paper. So this is personal publicity. It took me about 1 year to write it. It is some 100 pages. So at least until 2022, you can find a sort of selection of relevant literature. And it is open access. So you can just go on the website and download it. So it is essentially cardiovascular fluid mechanics from the point of view of fluid dynamicists. So it is easy to read if you have a little bit of competence in fluid mechanics. Okay, now we can start with the problem. So why the human heart is important? Now, at this point, I have a collection of different motivations, depending on the audience I'm talking to. When I go to medical doctors, I can talk about the money spent from the healthcare system for cardiovascular disorders. But since all of you are scientists and computational scientists, I don't need to bother you with the budget, with the cost, and I can just bring you to the essence of the problem. So the heart is really something fascinating. When you study the numbers, it is something you hardly believe, because it is 0.5% of the human mass, and still it can achieve something like feeding the systemic circulation, which is 100,000 kilometers long. Then it runs only on 8 watts, which is, for those of you who are engineers or have a little bit of knowledge about what a watt is, it is a ridiculous power for a pump. And it is even more surprising if you realize that just 1/4 of this power is used for pumping the blood. The rest is used to make heat. And this is important because the blood is not only pumped, but continuously warmed by the heart. And well, it can change by a factor almost 50 the available power. So when you make an effort, you can go as high as 300 watts. And then what is really surprising is that it beats for 40 million times per year lifelong. So if you have, if you are lucky and if you have an healthy lifestyle, all lifelong you can keep your heart, which keeps beating all life. And this is something which triggered my attention, because at some point I asked, what are we doing compared to the other animals? So are we special? Are we just like the other animals? And so this is something I have digged out from several papers. So here there are, initially it is naked, but now this slide will be populated. So maybe I better use the pointer. So HR means heart rate, and LS means lifespan. Okay, so let's start from here. So the heart rate is beats per minute, and lifespan is years. Just to make things comparable, I've collected data for warm blood mammals, because if you take fishes, reptiles, birds, then they have a different circulation. They are different kind of systems. But these one are all mammals, so they all have a complete circulation, meaning that there are in fact 2 circuits. The heart is made by 4 chambers, so we are comparing really apples with apples. Okay, so you can see here that there is a sort of correlation in the sense that hearts beating faster are those who last less years. And this is somehow expected. And here you can see lifespan compares to heartbeats. Okay, so this is essentially the number of beats per minute multiplied by the number of minutes during a lifespan, And then you get the number of beats of the heart during lifetime. So you can see that all animals are around a sort of cloud. So this means that biology has evolved the heart to beat a finite number of times, which is about 1 billion, for all mammals. And now we are putting some names, so some animals. So you can see the rat, which is very small, has a heart beating very fast, but unfortunately it lasts, say, just a couple of years. On the other hand, you can have a whale, which is a huge mammal, and then the heart of the whale beats very slow, some 20, 30 beats per minute, and the lifespan is some 30 years. And then you can have in between, say, dogs, lions, and so on. But there is an outlier, and the outlier is man. So man, as you can see, is completely out of scale. It's a unique case, essentially because for our heartbeat, which is around 60, 70 beats per minute, we should live something like 20, 25 years, which is very depressing as a perspective. And the heart, which usually stays for 1 billion heartbeats during a lifetime, for humans, it is 3 times more. So a question is, are we blessed by some God, or have we made something which has changed our fate, our destiny? And this is the interesting part of collecting data and trying to understand. So this is the Neanderthal man. So the red bullet is what happened when man had done very little to change the environment around. So Neanderthal beings were living, in fact, as biology would predict. And then things started to change. So this is the Neolithic. And this is a classic age. So classic age is at the times of Greeks and Romans. And then medieval age. And then early 20th century. And this is the current age. So as you can see, our progress in changing the environment, cooking food, living in comfortable environments has continuously improved our lifespan. I should mention that there is huge uncertainty on these numbers. If you put error bars, these are huge because you can imagine collecting data at classic age. But we only have the data for people who somehow have left a trace in their life, and then people who probably were wealthy, were living in conditions which are not the standard ones. But anyway, the trend is clear. And so what is the conclusion? The conclusion is that from a biological point of view, humans are just common mammals. So we are nothing special, but thanks to our capability to change the environment and to improve our conditions, we have been able to extend our life expectation. And then this is a good motivation for studying the heart, which is not what has driven me since the very beginning, but this is a good reason for keep studying the problem and seeing whether it is possible to extend further this life expectation. Okay, now what is needed to study the art? Okay, now if the title were not covered, it is really annoying. Oh, do I? Okay, it was. Probably. Hello? No.
So let's
Now it should work. Let me close. I will put here. Okay. Now, what do we need to study the problem of the heart? Well, we need a lot of ingredients. And as I'm writing here, each one is a tough problem in its own. So let's try to look at the list of ingredients. So first, we need to define the geometry. So how the heart is made. And this is already a difficult question because the heart is very complex. It has chambers, it has deformable tissues, it has intrinsic properties, and this is really the tough part. And most important, each one has a different heart. Even among healthy people, each heart is different from the other. So it is impossible to define a single heart. Anyway, once you come up with a geometry, then you have to find a way to solve the flow inside this geometry. And for those of you doing fluid mechanics, I assume most of you already know how difficult it is computing a flow in a complex geometry, like the heart is. On top of this, you should put the fact that the heart is moving, is deforming, and the motion depends on the hydrodynamic loads, among other things. So it's not something which you can define on a steady, complex geometry. And since you have also the elastomechanics of the tissues, then you should have also something solving the structure. And the structure undergoes large displacements, which is quite a difficult topic for those doing structural mechanics. And the tissues are not linear. So they are not elastic linear materials, but they are hyperelastic, anisotropic, non-linear. So all bad things are together. Then there is fluid structure interaction. And then there is a new ingredient, which is the electrophysiologic system. We will see that for the heart, it is the orchestra conductor. It is the system which determines who is contracting, who is relaxing. And then everything must be done, must be implemented within a numerical solver. And if we want the solver to be used by medical doctors, then things must be done within the time of a diagnosis. So if you take 6 months to make a good simulation, well, maybe you make the cover of a journal, but you cannot go to a medical doctor and say, I have something simulating the heart. You would never be considered by doctors. Okay, so this is the framework in which we had to develop our model of the heart. So let's start from the geometry. So the heart, well, it has quite a standard structure in terms of suborgans, because there are 2 atria, which are 2 small chambers. Then there are 2 ventricles, which are the real pumping parts. And then there are valves, heart valves, 2 separating atria and ventricles, and 2 giving the right direction of the blood to the systemic and pulmonary circulations. And then there are veins and arteries, which are attached to the heart, and they are key to prescribe the right pressures to the heart. Now, the heart is extremely specialized. So every part of the heart has different myocytes, which are the cells of the tissues, different thicknesses, different mechanical properties, different electrophysiologic properties. So if you want to make a reliable model for the heart, you must account for these highly specialized tissues which compose the different parts. And this is not enough, because I had mentioned that the myocardium, so the muscle of the heart, is anisotropic. What do I mean by anisotropic? The tissue is formed by fibers, and these fibers are aligned along precise directions. This goes back to our fetal development. Initially, it is just a small sheet of tissue which starts folding around precise directions to form the heart. And all a myocyte can do is contract and relax, okay? And so it is only because of this particular orientation of the fibers that the heart can contract along precise directions with different timings. We will see that there is a twist of the heart while contracting, which is key to properly squeeze the heart from the chambers. So when you model the heart, you cannot just use an homogeneous material. It wouldn't work. It wouldn't be a reliable model. And if you really look at the model, not only fibers have precise directions, but they have even different directions across the thickness of the myocardium. So those of you who know composite materials, you might know that fibers are aligned along different directions just to give strength other than the individual strength of the fibers. And nature has done it, well, ages ago. Okay, so we had to develop a long story, a long expertise to make the heart. But then another question comes, which heart do we simulate? And now there is a bifurcation. There are two possibilities. We can take the heart of someone, for example, my heart or his heart. You take medical images, you try to be as detailed as possible. Or what you can do is to take a sort of average heart. Okay, so you take, say, 100 medical images, you make averages, you take data from medical atlases, and you try to make a standard heart. So what is the difference? Of course, every approach has advantages and drawbacks. If you take a single heart, like mine, for example, in principle, you are doing the analysis for one specific organ. And so if you give an answer, that answer applies for that organ in particular. In the other case, when you make averages, you provide answers which could apply for all possible organs, but for any of them in particular. And then it would be more correct to, rather than making a single simulation, you should make a simulation campaign in which every parameter, which you know by the average, is randomly varied with some probability distribution of the uncertainty. So this is called uncertainty quantification analysis, and this is something we have done here at GSSI with one of the former PhD students. So we have decided to take this second possibility on several counts. One might be a little bit unpopular, but it is something I strongly believe. Patient-specific analysis, which is what you do when you take a particular image and you do the analysis on that, on the heart, is not possible. It's not possible because the geometry is always idealized. When you take an image, even the most detailed CT scan you can take, you are always smoothing out a lot of information. And also because there are quantities which you cannot know, like the electrophysiologic tensor or the electrophysiologic system, or the mechanical properties of tissues which, when they are alive, you get something. When you explant and measure, you get something completely different. So initially we have tried, but after a while we were really frustrated by the impossibility of getting data which could make any sense. So eventually we decide to take the other direction. Oh, okay, so this is a real art, and this is just to give you an idea how a left ventricle is made inside. So no matter how detailed is your CT scan, you can never get to that detail. The reason is that the amount of detail you can get in a CT scan depends on the radioactive dose you give to the patient. And of course, there is an ethical limit you can use to get details. You cannot kill the patient just to get a very good geometry. And even if you did, still you wouldn't know about electrophysiology, mechanical properties, and so it is essentially hopeless. Okay, so this is a little bit of additional information. So these one are all healthy hearts, and you can see how different they are. This is a single heart, but the imaging is made using different techniques. And so you see that the final geometry which you come up is different. So this is just to reinforce my view. Well, our view, actually, we are a group. Okay, so let's pretend we already have a geometry of the heart at hand. Well, we are ready to start with the model, okay? So the model, as I mentioned, needs three ingredients: fluid, structure, and electrophysiology. So how the fluid looks like, this is the most familiar part for me, because it is Navier-Stokes equations, and this is the gold standard for any fluid dynamicist. Here there is a little bit of ambiguity about the viscous stress tensor, because blood is not a Newtonian fluid, it is a shear thinning fluid. And so rather than using the standard Newtonian constitutive relations, there are some elaborations of it, like Carroll-Yasuda shear thinning, but this is really a minor point. It is well known what to do with this. By the way, the code, the numerical solver for the fluid is this one, www.afid.eu, which is a Navier-Stokes solver which we have open sourced a few years ago. So it is very much likely that you will put your hands on this solver at some point of the school. But anyway, just go there, download, there are tutorials and all sorts of things. Anyway, this is the Navier-Stokes solver, but then we have a point, which is how do we deal with the complex geometry of the heart. And this is one of the key ingredients of the model, which is immersed boundary methods. Now you will have an additional few hours of me next Friday talking about immersed boundary methods. So I will show you the details and all the tricks behind. So for the time being, it is enough to say that it is a numerical technique used to simulate flows in complex geometries without having a body conformal mesh, which for the heart is unavoidable, because already making a mesh for a steady heart would be difficult, but for a moving heart, it would be impossible. And here I'm showing you why. One of the bad features of the heart is that the fluid domain changes topology during the heartbeat. So when the valve is open, atria and ventricles are a single volume. So in principle, you could mesh in this way using a single domain. But when the valve closes, the two domains are separated. So atria and ventricles make different chambers. And all the coordinate lines here would become singular because they would collapse onto a single point, a single surface in space. So this is not difficult. This is impossible to compute unless you play tricks like leaving the valve a little bit open, and then you put some sort of numerical momentum loss to prevent the leakage between the chambers, but it is something which we prefer not to play. Anyway, you will hear more about this on Friday. Okay, this is for the structure. Now, the structure takes into account all the rheology of the elastic tissues, and it has several different models within the same solver, because there is the myocardium, which has a thickness, so the model must handle three-dimensional tissues, but the valves are thin membranes. And so the model has to cope at the same time with two-dimensional thin structures. And everything is taken into account within the same solver. Of course, we have the constitutive model, which is different for myocardium and membranes. And you can see here that valves can be simulated with tiny details. And this is crucial for the heart, because the flow going through the valve enters the heart chambers. So I'm saying this because there are many heart models on the market, meaning in the literature, who do not include the valves, and they just have sort of momentum loss volumes, which of course can work to open and close volumes, but cannot reproduce the flow downstream of the valve. So in this case, there is a biologic valve on the left. This is an aortic valve, free leaflet. And on the right, there is a mechanical valve. So this is a prosthesis well, hopefully not, but a prosthesis a patient could get in case he or she must undergo valve replacement. And you can see how different is the flow behind. It is really completely different. Okay, now the electrophysiology, and this is unexpectedly the most difficult part, because it has completely different time scales with the fluid and with the structure. And so where this model comes from? So the myocardium activates because an electrical signal propagates along the myocardium, but it is not just a medium in which electrical waves travel. There are insulating parts, there are very fast fiber connections, and this is because all the system must contract and relax with very precise timings. Now, what happens for a single myocyte, for a single cell, it is well known. There is essentially a stream of calcium ions going through the membranes, which open and close gates, and then the valve contracts and relaxes. So at the level of a single cell, models are extremely well developed. The problem is that there are too many cells in the heart to simulate the whole heart starting from single myocytes. Just to give you a number, in the left ventricle only, there are 5 billion of myocytes. So making the whole heart from myocytes is out of question. And then there are effective models. Now, this bi-domain system of equations might look a little bit weird, but it is nothing but a generalized Ohm law written for an extracellular medium and an intracellular medium. And then there are connections in between. It is important here to see these two lines, because what you get here is the red line is what the electrical potential does for a single cell. And each cell, well, each region of the heart has different cellular models. And the blue line is the mechanical tension, the contraction of the cell. And so we should solve this system of equations all over the heart. But things are more complex than they look, because as you can see here, there are thin one-dimensional, say, highways for the electrical signal. And over here, the signal can travel as fast as a couple of meters per second. Then there is a sort of gate here, the atrioventricular node, and over there the system is delayed by 100 milliseconds. So the trick is that the atria contract, they push the blood down to the ventricles, but the ventricles do not contract. Because the point is like having a balloon. If you squeeze the balloon in all directions, the fluid inside doesn't go anywhere. So the trick is pushing in one direction so that the fluid moves down, and then pushing the other direction so that the fluid goes away through the valves. And all this is taken care by the electrophysiologic system. Okay, and here there are the main ingredient, the main systems with different cellular models. And so here you can, well, on the left, it is just an animation, just to show you how the system looks like. And this is our one-dimensional bundles. This is a sort of two-dimensional network called Purkinje fibers. And over here, the system can travel around 4 meters per second, so really super fast. And then there is the myocardium, where the signal travels 50 centimeters per second. And it is all these different timings and delays which gives the proper contraction of the heart. Well, different cellular models. Now we have all three systems, okay? In principle, we should be ready to solve them, but we are not there yet because the system must be coupled. Okay, so you have the fluid, you have the structure, and fluid and structure work together because the fluid determines the hydrodynamic loads on the tissues, and the tissue, by active contraction, determines the loads on the fluid to be moved across the systemic circulation. Things are a bit more complex than this because the electrophysiologic system is coupled to the tissues through the local mechanical tension. The speed of propagation can change when the mechanical tension changes. And so all this must be solved together. Well, if this is not enough, there is an additional problem, which is boundary conditions. So in fact, the real circulation does not have boundary conditions because it is a closed circuit. But we do not have the computational power to simulate everything. And so we must chop, we must interrupt at some point veins and arteries and put artificial conditions to mimic the effect of all the missing part on the system. Okay, and this is important because the system has inertia, has drag, and then this must be done using sophisticated boundary conditions. I will not spend time on this because it would take an additional seminar about how to make this boundary condition work. Well, once you have everything, you ask, how do I validate? And this is a very difficult question for this problem because if you have a simple flow, what is called a canonical flow, like flow in a channel or turbulence, it is very easy because you have a neat geometrical configuration, a neat system of equations, and then you have benchmark results which anyone trusts. But in this case, well, if you take data from humans, as I said, each one is different. And then you can never know whether you are just making colorful pictures or reliable data. So we have followed two different directions. One is run in-house experiments. So we have developed a lab with experimental setups inside so that we could develop part by part and benchmark what we have done. So the part for the valves, so the part for the ventricle, and so on. And then we have taken biomedical data. We have averaged like medical doctors would do, and then we have verified whether our data were within the average medical database. So you can see here, for example, for clinical data, our results are the red lines. Now, whatever they mean, they are twist of the ventricle, contraction, semi-axis. Well, they compare reasonably well. This is something which is not spectacular, but we are very proud of. This is called the Wigger's diagram, which gives the pressure in the ventricle in the various parts of the cycle. And it is not something we have tuned. It is just the result of the simulation. And this is the experimental apparatus. I don't know if you can see here. Yeah, you see there is a ventricle inside. And what is nice is that this is something we can produce ourselves using isotropic materials of which we know perfectly the mechanical properties. And so we can make comparisons with the code. So you can see here, we have seeded the flow. We have performed the PIV PIV measurements. So this is how we make ourselves the ventricles, how we produce them. And then we make comparisons. So you see here, so the yellowish picture are experiments, and what is on the left, on the right, is numerical simulation. Well, this is closing. Well, there are several comparisons, but okay. So let's assume that you believe that all the comparisons are satisfactory. Then we can start with the simulation. And now there is no cheating here because data are those provided by nature. So there is not much we can do. The heart has a given dimensions, the blood has a given viscosity. And then we used exactly what is the data set for a real heart. And the bad news is that in order to have reliable simulations, reliable results, the number of grid points we had to use to simulate the flow was in between 200 million and 1 billion, because essentially the Reynolds number is what it is. And if you don't want to cheat, you must resolve the smallest flow scale you generate, which is the Kolmogorov scale. In this case, it is some 30 microns. So you must tessellate a volume which is 15 centimeters each side in a cube by subvolumes, grid cells, which are 30 microns in size. What is even more discouraging is that you have three systems. Each one has its own dynamics. And most difficult, as I said, is the electrophysiology. In order to properly capture the electrophysiology, you must advance in time your simulation with time steps which are 2 microseconds. But the heartbeat is about 1 second. So it means 500,000 of time steps per single heartbeat. And you must simulate several heartbeats because you want to make averages. The cardiovascular problem is a bad problem from the computational point of view, also because 1 billion grid points might look a lot, but it is not very much for those doing turbulent simulations. Now we are running simulations, say, with 50 billion grid points, 100 billion grid points, it can be done. But of course, it must be done when it is necessary. And using so many grid points allows you to use an equally large number of processors. But here it is not. Here you have, say, few hundred billions of grid points, which you can exploit by, say, 100 processors. Using more doesn't help, because then the overhead by communication outperforms what you gain in speed. So you cannot fully exploit supercomputers just because the problem is not large enough in terms of size, but you must solve using very small time steps. And so this is what we got initially, which was extremely depressing, because what we could do was to run one simulation, which is the initial transient, then some 10 heartbeats in 3 months. So 3 months is, say, a flagship simulation, something you can put in the cover of a scientific journal, but it's not something you can use for medical doctors. They would never pay attention to you. And so we really didn't know what to do. But this is why the serendipity of science is so nice. Now, let me move this window, because I really like showing the picture of these two guys. So this is Josh Romero, and this is Massimiliano Fatica. Now, Massimiliano Fatica is a close friend of mine, and we did the PhD together, sharing the same desk. Okay, so we were not roommate, but really desk mate. And he went, well, he did many things, and eventually went to NVIDIA Corporation, the one doing graphic cards. And he was one of the first to realize that these graphic cards, which nowadays go under the name of GPUs, could have been good for intensive scientific computing. And so we discussed with them the problem. And they said, well, we know how to do it. It's very funny. They are really nerds, but in the good meaning of the term. And so they really wanted to help us, and they helped us with the porting. And so they came up with something which we could not recognize. So we could not see what we had programmed at the very beginning. But the good news is that what initially took 3 months eventually was reduced to 1 day. So essentially, one full simulation could be run in a single day. And this was fantastic, because now we get down to the time we can use to talk to medical doctors, because now it is something that we can discuss. And then we opened really a huge variety of possibilities, because now we could really use this tool as something to be employed for clinical research. And so something we decided to do was to use this code for virtual clinical trials. So rather than running a single simulation, as I said, we could have run a simulation campaign of, say, a few hundreds of different simulations. And this is nothing but what medical doctors do. Whenever they come up with a new procedure or a new prosthesis, what they do, they apply to a cohort of patients, and then they make statistics and see whether the new idea is better than what was already available. And now this idea was appreciated so much that Francesco, who was sit there, got an ERC starting grant on this project. So it is really something valuable also from the scientific point of view. So now I will show you briefly a few applications we did for this model. So one of the first application was the cardiac resynchronization therapy. I mentioned that the electrophysiology of the heart is key to keep everything synchronized. Now, something that can happen is that one of the fast conduction highways in the left part, for some reason, becomes disconnected. This is typical when you have an ischemia, which is essentially an infarcted area. So fortunately, nature has been very careful in this respect. Even if this happens, the heart can still beat, although not with the optimal synchronization. Okay, so the left ventricle still beats, but a little bit slower, just because it cannot rely on the fast conduction highways, but it must rely on the slow conduction of the myocardium. Now, when this happens, the performance of the heart decreases, and then medical doctors can implant a pacemaker. So essentially, a pacemaker is a small box with 3 electrodes. One senses the heartbeat. The other one, well, there are many models. The one for this pathology has 3 electrodes. The first one in the atrium senses the pacing of the heart. The second one on the right feels when the electrical signal gets there. And the third one, which is the active, whenever the second gets the signal, sparks a new signal from the other side. So if you think it is a sort of bypass to the damaged electrical part of the heart. Now, there is a problem. The problem is that the pacemaker is implanted through veins, not through arteries. So the veins can reach the right part, while the damaged part is on the left. Okay? So what medical doctors do, they make some opening here, and they pinch the vena cava, which is the largest vein we have, and then they follow the path until they get to the right atrium, which is this one. And then going through the tricuspid valve, they go to the apex of the right ventricle, which is this one. But then they must reach this point. Okay? And this is the left ventricle. So what they do, they go through the venous system of the heart. It is called coronary system, which is not coronary arteries. It is a different system. And here there is a problem that sometimes these veins are very tortuous. Sometimes they don't get to the right direction because it is a sort of trees which they have to go back. And then what they do, they put the electrode at the best of their capability, but at somehow a suboptimal position. And so we had the idea of making a sort of clinical trial to see what is the outcome of the surgery depending on where you get the implantation. So what we have done, we have started from an healthy heart. Then we have artificially induced a pathology by disconnecting the electrical connection. Numerically, it is very easy. And then we have reimplanted the electrode in the impaired part. And here I can show you some of the results. So you can see the electrophysiology, and you see that here, where there is the pathology, the part does not depolarizes, just because this part is not reached by an electrical signal, okay? And you can see here that there is one part which is not depolarized at the right time. And you can see that this has influence on everything. So this is the mechanical contraction. Now here it is a little bit more difficult to see, but if you isolate the right frame, you see that the impaired heart as the left side, which is this one, this part we are looking from below, which does not contract at the right time. So during systole, which is the part when all the blood, all the heart, the ventricles should squeeze, the left part doesn't do it at the right time. And also the fluid dynamics is a little bit tricky to see, but if you isolate the right frame, you see that there is a systolic jet. So this is in the aortic arch, which is physiologic for the healthy heart, restored for the heart with the pacemaker, but it is very weak, almost absent for the impaired heart. It will come a little bit later, but it isn't good when there is not the right synchronization. Now, of course, you cannot tell medical doctors about contraction, about jet velocity. It is not the right way to communicate with them, but pressures they understand very well, provided you convert in millimeters of mercury, because if you talk about Pascal, they don't like it. And so you can see here, the black part is the healthy heart, so the peak systolic pressure is 130 millimeters, and the impaired one, the one with the left bundle block, loses almost 30% of each peak pressure, and this is reflected in all other performance. The most important one is this EF, which is ejection fraction. Essentially, it is the percentage of blood in volume you squeeze every heartbeat from your heart. So a physiologic value is in between 45 and 60%. Everything below 45 is pathologic, below 35 is life-threatening. And so you can see here that with this pathology, you get to 34, which is quite a worrisome number. And if the resynchronization by pacemaker is properly performed, you recover 48 out of 51, which was the original value. What medical doctors really like is ECG, so electrocardiogram. And since the model does everything, including the electrical signal, we have put our heart in a model of a human torso, and then we have collected the electrical signal propagating at the human torso in the position when doctor typically put the electrodes. And so we have built synthetic electrocardiograms. And we have checked with our surgeon. So if you, it is unlikely, but at the very beginning, there was a list of people collaborating with us. And one of them is Ruggero De Paulis, a very famous heart surgeon. And so we have shown him these ECGs. And apparently, they are very realistic. So what he has said is that this part, when it is small in amplitude and of longer duration, indicates really a mismatch of the electrical conduction. And this is the restored signal. You can see here this spike is the spark shoot by the device. Now, I don't recall the name. Pacemaker. Yeah, here it is the name. And now this is the trial. So what happens if rather than putting the electrode at the optimal position, which we know because of numerical simulation, you put in a different position, just because the cardiac surgeon cannot do any better without knowing in advance where it is the damage exactly. And so you can see that you get a wide variety, a fan of different possibilities, which like this are not easy to interpret, but if you put in a table, you see that this is the healthy situation, this is the impaired situation. This is the optimal situation, and these are all possibilities. Okay, so you can see that you can go from some slightly suboptimal numbers to something which essentially tells you that the surgery hasn't had any appreciable effect, just because the electrode has been positioned in a place where it cannot do much. Do I still have some? Yeah. Now I will quickly go through another couple of applications in which different aspects of the heart have been investigated. So one of the beauties of having a numerical model is that even if you have everything available, you don't need to use everything if it is not necessary. So although we have all the heart, all the chambers, all the valves, all the systems, there are problems for which only part of it is necessary. So this is a case which has recently been studied by Francesco and by other people. Some of them are really even here at this moment. So this is a problem in which you need only the left part of the heart. And in particular, it is the problem when you replace the heart valve. Where do you put it? We will see that surgeons have a little bit of freedom in the positioning of the valve. And then one can ask, where is it better, depending on the conditions? And this is what Francesco and the others have studied recently. So you can see here, this is an MRI of the heart, of the beating heart. And these two tiny structures, opening and closing, these are the mitral valve. So the mitral valve is essentially like a €2 coin. It has 3 leaflets, which open and close passively, just because of the pressure difference between the left ventricle and the aorta. And this valve regulates the flow from the heart to the systemic circulation, because from the aorta then all the body is fed, all the organs and etc. So the way it works, when the ventricle contracts, there is higher pressure in the ventricle, the valve opens and the blood flows through the valve. When the heart relaxes, and this is called the diastole condition, blood is pressurized into the aorta, whose walls are quite rigid but still elastic. So all the elastic energy goes back into the system. The blood tries to go back, but then the valve closes. And this is why in our model of the heart, we have to consider also the main vessels around. Otherwise, you don't get the right functioning of the system. Now, since the heart beats 40 million times per year, this valve opens and closes 40 million times per year lifelong, if everything goes well, because depending on some pathologies or some unhealthy lifestyles, this is by far the most the organ most subjected to disorders. One of the most common is that some calcium starts building on this valve, which becomes stiffer and stiffer. Then a reduced area can open and close, and then if the same flux has to go through a smaller area, velocity must be bigger, and then you have a lot of problems, like a pressure drop increased, which means that the heart has to work more to counter the pathological resistance. And then you have the blood who flows faster, and we will see that the mechanical stresses are a problem for the heart. At some point, medical doctors have to do something. They do as much as they can to replace, to restore, to spare the original valve, but sometimes they are obliged to replace. And there are many different models. Now, of course, the first two are just historical models. They are not used anymore, although the first patient who had this one implanted is still alive. So this valve is working for some 60 years without stopping working. But nowadays, essentially, there are three possibilities. This one, which is a mechanical valve. This one, which is called the biologic valve. And these ones, who are the latest possibility, these are essentially the same as this biological valve, except that the valve is enclosed in a stent. So rather than opening your chest, stopping the heart, opening the heart, and sewing the valve is inserted via the arterial femoral artery. Okay, so there is a catheter. It is inserted from here. It goes up through the aorta, the aortic arch, and then it is deployed at the right position, which is very appealing, but it has some drawbacks, because if you open the heart, you can clean very well and take out all the calcified part. If you have to open this valve within the impaired one, of course, you can just push aside the deteriorated part, but you cannot eliminate. Okay, so there are, of course, a drawback, but of course, there are patients so elderly and so with such a bad health for which stopping the heart and opening would be inconceivable, and so they can benefit from this new technique. So just to complete the description, is it better to have a mechanical valve or a biological valve? It depends on many different things, because the mechanical valve, it is a single implant in a lifetime, because the valve lasts more than 80 years. So you get only one implant. But since it changes a lot the flow, you must take anticoagulants for good. It is a lifelong therapy. And so you can have hemorrhagic events. So there are some drawbacks. The biologic one, which is this one, it is made by bovine pericardium, so essentially cow, the sac which encloses the heart of cows. It has an hemodynamic which is very similar to our native valve, and so you don't have to take any anticoagulant, but it is advertised for lasting some 15 years, but in fact, after 10 years, you must make a replacement. So if you are very young, it is very much likely that you get a mechanical valve. If you are, say, around 75, then you definitely should go for a biologic valve. If you're in between, well, it's up to you. It is a tough choice, and you are asked, actually, which one would you like? And so, yeah, it is a difficult choice. Okay, so this is the point. Where do they, surgeons, implant the valve? Well, nature has put the valve here, so there is an ostium, but the real valve is just leaflets. The prosthesis is not just leaflets, because there is a frame, there is something in between. So whenever you put a valve of the same size as your ostium, you don't get the same orifice area. And so for those having already a small orifice, it might be a problem to get a valve, a prosthetic valve. Yes? Okay, so it is like shoes. There are sizes. They range from, say, 17 millimeters in diameter up to, say, 27, more or less. There are even bigger, but they are implanted in the mitral position. So it is a different kind of surgery. So usually what medical doctors do, they make ultrasound imaging. So they take a measure, and so they have an idea of what to put in advance, but they have anyway during surgery available the set of different valves so that, and they have a device. So they put in the ostium, they take a measure, and then they decide which to put. But in some cases, for example, if someone has a very small body, putting a prosthetic small valve might be a problem because the available area is too small. And then the surgeon can decide to implant the valve in this position, which is above the ostium. So they know by experience that it is better. But then, thanks to the numerical model, we could see why it is better. So it is better on several counts. Okay, so this is what I was saying before. Actually, the size is what you get from here to here. And for the native valve, it is all available space. But when you implant the prosthetic valve, you have much reduced ostium for the blood. Okay, so these are the possibilities. Okay, and this is the model. So the model needs only the left part. So there is the mitral valve, there is the prosthetic valve. There are these sophisticated boundary conditions to get the appropriate pressure differences. And then here we can impose volumes rather than computing the time-consuming electrophysiology. Okay, so something we haven't mentioned up to now is the problem of hemolysis. So what is hemolysis? Blood is not water. So water, you can do whatever you like, you don't damage water, but blood is very delicate. It is a very dense suspension of cells, living cells, who are very sensitive to mechanical stresses. So whenever you change your valve, you change the mechanical stresses these cells are subjected to. And so one of the problems when you put a prosthetic device is that you are altering the mechanical stresses which act on the cells. Now here I have added some old slides. So this is the blood seen in details, and this is the kind of scales you get. So you get large scales, but you get very tiny scales in turbulence. Okay, and then what happens to the blood, you can see here. So the low shear levels, red blood cells make a sort of structures called rouleau. By electrostatic forces, they tend to join. When the shear increases, they tend to tumble, but they behave like solid body objects. Then when you still increase the stress, there is a phenomenon called tank treating. So the phospholipidic membrane starts to shear and the fluid inside rotates, but this is not a problem because red blood cells do not have the nucleus inside. So still you can, still the cell can resist, but if you still increase the mechanical stress, you tear the membranes and you have lost for good the red blood cell. Now, this is not a problem for the red blood cell itself. We have many millions per cubic millimeter. But once you open the membrane, you release the hemoglobin in the plasma. And hemoglobin is toxic for kidneys. So it shouldn't go into the plasma. The other problem is that the same mechanical stress who tears the membranes activates platelets. So platelets become active tend to grab these damaged cells and form clots. And these clots start circulating and then can create strokes or ischemias. So it is really something it shouldn't happen, which is the reason why mechanical valves go with this lifelong anticoagulant therapy. And so you can see here, these are sort of particles released in the bloodstream. And you can see here, this is what happens to a single mechanical, to a single red blood cell during the path. Now, this was a small system, and the mechanical dose to each red blood cell could be computed following the path. So it is called the Lagrangian tracking. But there is a more clever way to do it, which is looking at the hemolysis in Eulerian way. So you can see the flow here with the two valves. Now it is very difficult here to understand what happens. The reason is that the flow has such a three-dimensional character that cutting a plane through any direction doesn't give you the picture. And then numbers can be collected. Okay, so you can see here a little bit of numbers. So GOA means geometrical orifice area, EOA effective orifice area, and TPD transvalvular pressure drop. So this is quite mechanical quantities which are very important for the circulation. And you can see that the supra implantation has always more attractive numbers. So you have a bigger orifice area, and what is important, a smaller transvalvular pressure drop. And this is really key because this is an overload for the heart, because the heart normally has to work to win the dissipation from the capillary bed we have all over the body. And usually in the valves, the pressure drop is, the medical doctors call a single-digit drop, means that the pressure drop is below 10 millimeters of mercury. When you have a prosthetic device, as you can see, numbers are much larger. And these numbers are, in fact, the difference between these numbers and the biological pressure drop is additional work for the heart without any advantage for the stream of blood. Well, there are many diagnoses, but this is important because this is the mean hemolysis. Okay, now HI means hemoglobin index, which is how much hemoglobin you are releasing into the plasma. And you can see that just by changing the implantation position, you can get an advantage of more than a factor 3. So this is something very important for medical doctors. And as usual, since we want to be sure that the numbers we are producing are not just fancy numbers, we run companion experiments to cross-check. So this is the production of the aortic root. So this is a 3D printed model, and then the silicon is painted around, and then it is polymerized. And then we have learned how to implant valves, of course, on models, not on patients. And see, and this is the PIV experiment. Well, these are some measurements on the flow. This is the transvalvular pressure drop. And as you can see, the advantage is always there. The green line is for the supra implantation. And it is the advantage becomes more important the higher is the heartbeat. Okay, so it is something which really counts. 5 minutes. Yeah, this is the last example I wanted to show, because before we have used just the tissue and the flow. Now, one example in which we show only the electrophysiologic system. And for a quite dangerous pathology, which is the ventricular tachycardia. So it is a ventricular fibrillation, essentially. So when the heart goes unsynchronized, it can be unsynchronized for the atria, which is annoying but not life-threatening. Most of elderly people have this pathology. But when it happens for ventricle, it is really life-threatening because the ventricle starts pulsating at high frequency, and it becomes incapable of pumping the blood into the systemic circulation. So this is really something you can die of. And so you can see here, this is a high-resolution left ventricle with the physiologic depolarization. Something we have not mentioned at all because it would have deserved another seminar is how the myocardium itself is fed by the heart. So there is a third circulation, which is the coronary circulation. So from the Valsalva sinuses, which are at the root of the aorta, there are two tiny arteries, a couple of millimeters in diameter, which are called the coronary arteries. And they branch through the myocardium, and they form this sort of tree, okay? This tiny system takes about 5% of all the blood pumped by the heart. So the myocardium is really eager of oxygenated blood, and it is a kind of tissue that cannot stay without oxygen. Whenever it happens in a few minutes, it is permanently damaged, and this is the cardiac ischemia. So this is what can happen. For example, at some point you have one obstruction, say for one of the clothes, which before we have mentioned. You could be so unlucky that one of these clothes, rather than going into the large aorta, enters one of these coronary arteries. And then since they keep decreasing the diameter, at some point they obstruct the vessel, and then all the downstream region is not perfused anymore by oxygenated blood. Now, there are some secondary perfusion paths, but when these branches are the main ones, there is very little that can be done. And then what you get is a patch of dead tissue, okay? So this is a necrotic part, which is a problem on many different counts. So first of all, it is a part which cannot contract. It loses the capability to contract. It cannot relax. It becomes very stiff. But what is worse, it loses the capability to convey the electrical signal. It has different conduction properties. And then you have the scar, which is the necrotic tissue. You have a region surrounding the scar, which has also different properties. And then you have the healthy tissue, the one who has remained perfused. Now, of course, depending on where the ischemic attack occurs, you have all possible outcomes from complete recovery from up to death. And one of the possibilities is that the scar is such that not only it prevents the electrical signal from crossing the scar, but since the electrical signal has to go around, this scar, you create secondary circuits. And this is what creates fibrillation, because the signal doesn't travel properly along the tissue, but it starts propagating around pathological patterns. And something that happens to the cells is that after they have contracted, they have a refractory time in which they cannot contract. But then, if they get again the signal, they can re-depolarize. And this is the problem, because they are depolarized not by the physiological signal, but just by the spurious signal who has been traveling about the scar. And so you see here what happens. You see, the signal starts traveling around the scar, and you never get rid of this depolarization. So what is the treatment for this pathology? The doctors, the hemodynamists, just go with a catheter and they try to burn locally the tissue to prevent this recirculation of the electrical signal. But this is a very empiric procedure based on experience, because of course they do not have any idea about what is the path of this electrical signal. And so this is how people here have tried to help using numerical simulations. So in this case, the collaboration is with the Campus Biomedico, which is an hospital in Rome. And they can do a very sophisticated analysis, because you can see this sort of spider below. This is a catheter with an electrode at every end, and this electrode can measure can measure essentially an electrical signal from which it can reconstruct the property of the tissue where it is probing, and also the conduction velocity by some indirect calculations. And so you can see here how many measuring points you can get. These are actual real data. It is not numerics. It is some real patient. Well, this is how to relate the electrical signal to the properties of the tissue. And so once again, we have to solve the model, an effective model for the electrophysiology. You see here, the cellular model essentially tells us what a cell does when it is reached by the electrical signal. And before I mentioned that there is a different cellular model for each part of the heart, and there is also a cellular model for the scar. So even impaired tissues have a different cellular models. Okay, then the way things have been done was starting from medical images, then a patient-specific geometry has been constructed, and then it has been approximated by a three-dimensional geometry. Now, already here, this is not one-to-one with the patient, because the patient was only characterized by the endocardium, so the inner tissue in contact with the blood, and the tissue thickness point by point was not given. But anyway, this was the best that could be done. And then conductivity maps were reconstructed by this sophisticated technique, feeding our electrophysiologic model with the numbers. So now the point is, using this input data, which is essentially the local conduction velocity of the signal, does the model reproduces the fibrillation? This is essentially the question. Well, the answer is yes, because this is what you get from the numerical model. Okay, and so you see that without any further impulse from the scar, you never get rid of the signal. So the signal self-excites itself, and this is in fact the fibrillation. In the physiologic heart, the signal comes only from the sinoatrial node, which is on the right atrium on the top, which gives the pace of the heart. Only that one should give the signal, but if you have scars or other pathologies, then you can have these extra excitations of the tissue. And now we can exploit the power of numerical simulation. So with a numerical simulation, you can venture and test any sort of surgery and treatments, even those who are hopeless, things which you would never do to a real patient. But here you can try and see what happens. And so this is the in silico ablation. So what happens if we burn the tissue? And we should see that at some point the signal dies out. Yeah, maybe it's not here, maybe later. Oh yeah, here it is. So you see that this is what happens when you do the ablation through all the thickness of the tissue, which is called the 100% ablation. And this is effective because you see that there is some unphysical circuit, but at some point the ventricle becomes all blue, which means that there is no self-excitation, while the other one, which has an ablation only through part of the thickness, you see you never get rid of the circuit. And now one might ask, well, why doing only a partial ablation? Well, the reason is that partial ablations usually do not impair the ejection fraction, actually produce less damage to the functioning part of the heart. Well, I think I've really abused of your patience, so we can go to conclusions. So I hope I have convinced you about the reliability of the system, and then what can we do with this numerical model? Well, other than entertaining ourselves, which is already a good bonus, but in fact, it can be used to test virtual prostheses. So usually, whenever someone comes with an idea about a new prosthetic device or a new surgical procedure, the point is that you start with animals and you see whether or not they survive. And then you use this procedure in hopeless cases. Now, it is bad to say, but there are patients which you know are dying in, say, 24 hours. And then you can test, you can try as really a last chance these new innovative procedures. And when you pass these tests, then you start the experimentation on a small group of patients, and then you go to large-scale clinical trials, and eventually it goes as a standard. Well, we believe that models like this could be used for the initial screening of the valid procedures. So, for example, you could spare animals just by testing virtually these devices on the model and see how it performs. And then you can study pathologies in clean conditions. Now, for those doing science, you know that running an experiment in conditions which are clean and reproducible is key. So you must be sure that if you do twice the same thing, you get twice the same results. Well, with humans, this is impossible. If you take a cohort of 100 patients and you do 100 times the same procedure, leaving apart possible mistakes, possible unpredictable events, but each of us is different. We have a different history, a different different quantity of an immunity system which is different. And then the same procedure on each of us would produce different results. So studying diseases on humans, it is difficult because you should always take into account variability. With a numerical model, you can change one factor at once and just assess the effect of that specific factor, which essentially is studying uncertainty quantification. Something we have done, we have a patent for this idea, is to extend the diagnostic capabilities of very basic devices. So here you can see the image you can get from an ultrasound device, which is very standard. You can do it anywhere. But then the disadvantage is that this can only give you a two-dimensional section. And how the medical doctor understands what is around by experience. He knows that when he sees on a section something, it is very much likely that what is around, the three-dimensional configuration, is something different. Well, then we thought, why, rather than using the experience, we don't use equations? So essentially, what we have done was to use the two-dimensional data from ultrasound to force the solution of the equations along a plane. And all the rest of the three-dimensional field is reconstructed by integrating the equations in time. So our idea is to partially replace the experience and guessing capability of the medical doctors with the predicting capabilities of physics and equations. So conservation of mass, balance of momentum, conservation of energy, everything is inside. Well, other conclusions are about the position of valves and the issues you can have in different implants, or here the possibilities that numerical analysis can do can give for the ventricular fibrillation. I think I will stop here. Thank you.
Okay, thanks, Roberto. And well, I would like to say that this is a really impressive presentation, and you know, you're showcasing how many things you can do actually with computers. And,
yes.
You have introduced at least one important keyword, which is GPUs.
Oh, yes.
Which we are going to deal extensively with in the next days. But for now, the floor is open to questions. We have
Really, Mike, okay. During the lecture, I didn't notice any strain tensor computation in this numerical method.
Sorry, any?
Any strain tensor computation.
Oh, yes.
Does it mean there is no section in this heart which is not plastic or plastic in long term?
No. So strain tensor computation, there is. Actually, there are many. But I didn't put all the details. So essentially, the way the tissue dynamics is computed is that you compute the hydrodynamic loads, so pressure and viscous stresses from the interaction with the blood. Then there is the active tension, and this is given by the electrical signal which contracts the myocytes along the fibers. And then the rest is done by the local mechanical properties. So it is the Fung constitutive relation, which is an hyperelastic relation. But it is there, of course.
Okay, thank you.
Actually, there is even more. Biological tissues are viscoelastoplastic. Something I've not mentioned, but since you ask, one of the reasons why the simulations take so long is that you cannot just start and get results, because the first, well, say, couple of cycles are needed in order to put all the tissues with the right tension. So if you take a strip of biological tissue and you start doing something like this, you see that the first cycles, you just condition the tissue. So you stretch to the right level, you produce the right elongation, and only then the tissue becomes sort of elastic. And this we do by discarding the first cycles. I don't know if I answered your...
Yes, I was just also wondering, like, if there is any section in HUD which is purely plastic, but in long term.
Okay, so plastic, I would say no, because plastic would imply that the tissue deforms for good. And this, at least in healthy situations, does not happen. But your question is very important, and I have not even started the point, but it is also another field of research. Something at which these models cannot be used is to predict what will happen, say, in 6 months or 1 year. Okay, because what we can do now is to simulate, say, 20, 50 heartbeats, and it is already a big effort. But then many times medical doctors are interested in if I implant the valve now, what will happen in 1 year? And so the problem is quite more complex, because what happens is that when you implant a valve, you change by a tiny bit, say, the local shear stress, say 5%. Now, immediately, nothing happens. But the abnormal shear stress at the tissue starts to trigger biochemical signals in the cells. And then the tissue from inside starts remodeling. So you get thinning or thickening of the tissues locally, just because the signaling time does not excite the system as it used to be in physiologic conditions. Now, this can be done with these models, but an extra module is needed, which is called mechanotransduction. So essentially what happens is that you compute a few cycles, you assess the new situation, then you go this situation to a model which predicts how the system will adapt, will remodel over the months. And then within the new configuration, you perform new simulations. So this is the closest I can imagine to plastic deformation, but it is called remodeling in the field.
Thank you.
Yeah, other questions? Yeah.
Thank you, Professor, for the presentation. So it's clear that the uncertainty quantification here, it's, I would say, the big still thing to solve. And so I would like to link this comment to one of the last slide where you show the patented forcing of the 2D plane measurements through the ultrasound inside your model. So I also would like to go back to the PIV. I see that still it's a planar PIV. So your model in the lab seems to be very, very a nice model. It's a small one. So I think that, like, for example, tomographic PIV or PTV, to have a 3D reconstruction of the field in terms of velocity, and then maybe go back to the pressure with some pressure reconstruction. I think that this would be a huge, at least starting point to have something to validate on. Are you considering this in your lab? Because it would be very nice to see.
So of course here I've shown only one PIV section, but since the system repeats itself over the heartbeats, we could change the planes, either vertically and horizontally. And so we have tested that over the planes, the system is reproducible. Of course, tomographic PIV would be a dream for us, but we essentially are not experimentalists. So we somehow were forced to run our own experiments just because we couldn't find anything to compare with. And we have made a lot of fun in doing experiments, but our real heart is in the scientific interest is in numerical simulations. We have also pressure probes, which we have put all over the system along the circuit, and we have checked. Of course, here I tried to squeeze in a couple of hours. I tried to put together a story and to squeeze some relevant results. So I could have focused on validation only, but it would have been very boring showing comparisons for a couple of hours. However, your initial question is absolutely right. Uncertainty quantification is by far the most important part of this research. Let me see if I have, because I had to skip a lot of material, but this is probably something I can show in a few seconds. It is also funny. So this nonsensical list of numbers are what medical doctors use mostly, and they are called nomograms. So they work in a very funny way. Essentially, you go to a medical doctor and he measures the pressure. So the pressure, it starts with a minimum, a maximum, and a minimum. And then he cast a segment, and you end up here. Now, this number doesn't mean anything. And then you cross with your age, which, by the way, is mine. This is just for fun, so almost 58. And you end up here. Now, this number gives you the stroke volume in cubic centimeters. And then you, again, cast a ray through your heart rate, and you get to the cardiac output. So when a medical doctor takes you measures, then he has either in printed form or in some electronic form tables like this. And for example, if you have to undergo aortic surgery, then they take a measure, and then they cross with your heartbeat, with your pressure, with your age, and they end up with a number, 4.5. And then they decide, well, you should undergo surgery. 4.35, no, you can still wait. This is the way they decide. So our idea was to replace these nomograms, so these tables, with something like this, which are called reduced order models, which are essentially digital nomograms. Now, for those of you who know a little bit, these reduced order models have to be tuned, and then in numbers. And these numbers are the number of numerical simulations you must run in order to tune these reduced order models. Now, how many simulations do we need? Well, it depends on the polynomial order you want to use for this model. The higher the polynomial order, the more accurate it will do the prediction, and on the number of uncertainty variables you have for your system. Okay, so these two numbers, P and D, are key to decide how many variables, how many simulations you need to tune your reduced order model. And here we have made some arithmetics. Now, I will make it very short. So if you take a polynomial order of 4, and you assume that the number of uncertain quantities for your heart are order of 20, then some 250 simulations are enough to tune your reduced order model. And this can be done thanks to uncertainty quantification, because by the knowledge of the probability distribution function of each quantity, you can sample each quantity in the appropriate way. Now, just for comparison, if you had to take random samples, so as would be for a cohort of patients, and they come as they come, you cannot tune the characteristic of a patient according to your will. You would need 30,000 people to do the same. Okay, and this, by the way, has been done by one of these former students of GSSI, Giulio Del Corso, who now is in Pisa at CNR, and he was really outstanding in doing all these statistics depending on the distribution. But indeed, uncertainty quantification is key in this kind of study.
Yes, just to curiosity one about the experimental model. How do you come up with it, and is it really representative of a, real model of a heart? I mean, it's just looking at it, it seems quite different, but of course you skip a lot because of time, but I'm really interested in how you came up with it. And the second curiosity is about, let's say, no, actually, no. The first one will suffice, I think, for the time.
Yeah. So your question is about the experimental setup. Yeah. Well, first of all, we have not reinvented the wheel. It is something which people doing experiments already know. But sorry, let me go back. So essentially, we wanted something which could produce a pulsating ventricle. Okay, so since I'm an engineer, I still remember all the topic I've studied, and so I realized that I needed something which was periodic, and so the idea was to have an electric motor which could be controlled by an inverter. And then this motor was connected to a cam, okay? So I don't know if you can see here, but there is a plexiglass shape. Now the shape gives you the time low for the piston. Now this is a piston, this one, and here there is a plexiglass box. This box is thick enough, a couple of centimeters, that does not deform. So the piston moving up and down with a given low determines the time low variation of the volume. And since everything is indeformable, the only thing which can move is the ventricle inside. Now, ventricles can be bought on the market. There is a Swiss company producing these ventricles, but they are charged in between 5,000 and 7,000 euros each. And they last, say, a couple of days of measurements. And so we have decided we better learn how to do it. Of course, we have broken some tens of ventricles, but then we have realized how to do it. Now, what is nice of this experiment is that it is done exactly in dynamic similitude. So the dimension is 1-to-1 with the heart. So we put the real valves inside. Since water is less viscous than blood, we can have the same Reynolds number at smaller velocity, which has also another advantage. We can go 4 times slower. So 15 beats per minute on this device is 60 beats per minute in the real heart with the blood. And then this is the old model. And now there are branches which we can test only for the aortic root or for other devices. And Francesco has downstairs another lab, and he's building another device like this for experiments here.
Do you also match the Weber number and the Reynolds number at the same time?
Pardon?
How do you do that? Yeah, like the surface tension and the Reynolds number?
No. Weber number, I never mentioned because it doesn't play any role here. Maybe it is Womersley number.
Yeah, perhaps that one.
Yeah. So the Womersley number essentially is the unsteadiness. It is the unsteady time compared to the convective time. Yes, it is a one-to-one also with the Womersley number.
I see, but then because blood is not the same does not have the same surface tension, I can imagine.
No, but the Womersley doesn't have the surface tension. It's essential. Well, I can show you. So the Womersley should be here. Yeah, here it is. This is, sorry, let me move this. Okay, so you see, this is Womersley. So D is one dimension. Nu is the kinematic viscosity, and the omega is the pulsation of the system.
So this is kind of like a Coulomb.
Yeah. And this, it is a close relative of the Coulomb-Carpenter number. Yeah, this is the kind of terms you must learn speaking with medical doctors. So in this community, Coulomb-Carpenter nobody knows, but Womersley, it is the common term. But it is more or less the same. Yeah.
There are any more questions? Yes.
I'll be there almost every day. So
yeah, just one quick curiosity. How skeptic are doctors when you show CFD simulations?
okay. So doctors are very fascinated by this model, but very difficult to convince to change their everyday routine. They have good reasons, because if they have learned how to implant a specific valve, changing valve implies restarting the training, and it is what they call a learning curve. They take some time, but in the while learning, people die. And so, of course, they are extremely conservative. To speak honestly, we have very few selected doctors with whom we can work very productively and very openly. And one of them is Ruggero De Paulis, this outstanding heart surgeons. Many others, we talk, we exchange ideas, but when it comes to putting things together, it is difficult. But initially, well, say 15 years ago, they didn't pay any attention. Now they ask, what do you think? What would you do? Can you run this simulation? At least they have realized that this is a valuable tool. Some of them really rely on the numbers. Some others are curious. I think over the years, it will be more and more that tools like this can become clinical tools. But yet the expertise, the personal ability of a doctor is by far the dominant factor.
Questions? Yeah. If there are no questions, I have a comment. I have a question. May I have a question?
Sure.
A comment is just a personal comment, because I think I saw the first time your simulation by the heart, maybe around 10 years ago in Cineca. I'm totally impressed by the improvement that every day, every year is done on this model. It's
You should look at there.
for Francesco. Is
No,
it some?
here.
Yeah, Francesco is not listening at all. Yeah, anyhow, it's impressive how the improving quality pieces, and you are essentially, let's say, modeling the most difficult tools that there is in life. Simply, that's the art. And the second comment is that in the last years, everyone in our research is talking about digital twins. Is a buzzword that I'm now hating a little together with AI. Okay, but apart from my personal, let's say, feeling, this is a digital twin. This is a real digital twin.
Yes.
Other are fake, honestly. Okay.
So digital twin is a word which I hate because it is so vague that it can fit anything. So this model is close to the heart. But for example, what happens if you take a medicine, say a molecule? We cannot do anything like this because we miss completely all the biochemical part. So we should agree on what we mean by digital twin. For example, a digital twin of a car doesn't need to have the same color of the painting or the same plate, but you can have a digital twin of the engine, a digital twin of the say mechanical structure. So this is as close as we can for the electromechanical part. But still there is a lot of biochemical processes, thermodynamic processes, which we have not accounted yet. So it is, I agree with you, much better than many digital twins claimed.
That is selling many proposal, paper and so on. But this could be hours of, let's
but
say,
we are very reluctant in using digital twin because it is far from reality.
Yeah. But it is abused. I don't like. Second question is, let's say, probably is out of context, but it's just my curiosity. Okay, what about if you started to change boundary condition for the blood? For example, you started to change the sugar concentration in the blood. So I'm talking about diabetes.
Oh, okay. So this is something we cannot do with this model. For the time being, the only part we can touch is the hematocrit, so the percentage of red blood cells, through viscosity. Essentially, we have almost a complete freedom on the way viscosity changes or the way viscosity changes with the local stress tensor. This can be done. But diabetes would be something belonging to the biochemical part, which would be interesting, but it needs different capabilities, different expertise. But it would be interesting, I agree.
No, I have a very stupid question. So I see you, well, computationally speaking, the main issue is about time step, right?
Yeah.
So and the time step is related to the electrical part. So my question would be whether you maybe can relax this limitation by accounting for the fact that different parts of your model have different time scales.
There are several tricks that can be played. One is if you relax the speed of conduction with the local mechanical stress, because in that case you can compute separately the electrophysiology, and then you run only elastomechanics and fluid dynamics. Of course, there are many things that can be done, but this, I would say, belongs to the partial use of the model. when we assembled the model, we really wanted to do something without any compromise. We wanted to do something which, for which we didn't have to apologize. And now that everything works and it's been reasonably validated, of course, we can play. So if we don't need a detailed electrophysiology, we can relax this and that constraint. But yes, there are many ways in which the system can be accelerated. It is also true that every year, faster GPU machines are released. And so what we needed 24 hours to run now can be done, say, in half the time. And it is very much likely that in 1 year or 2 years, the time will be halved. Maybe in a while, clean input data will be more an issue than CPU time, because that is really a problem. The group is really struggling with finding reliable geometries to make a cohort of virtual patients. That is really difficult because you find most of the hearts just for the left part, because it is the one undergoing the largest pressure differences. But in order to run all the model, we need all the heart, left and right. And medical doctors say, well, we cannot give radiation to the patients just because you need also the right part. And then valves are very poorly described, and without valves, we do not have the whole geometry.
Okay, so let's thank the speaker again. Thanks. Thanks a lot, Roberto. And I think we'll now